focusing on nucleic acids, and also we're going to be focusing on the building blocks of matter. Basically, we're going to take a deep dive into the nucleus, and in the nucleus we're just going to keep focusing on the DNA, which forms the building blocks of life, or the building blocks of proteins basically. So basically on your right there is the exercise that we're going to be focusing on, and then on your left is where I'll be demonstrating to you answers to these questions. Okay, I'm not a perfectionist, I might make mistakes. Please comment down below if I made any mistakes regarding the question. Okay, so the first question that we're going to be looking at is activity 1. So basically in this diagram we're given activity 1 DNA. So the diagram on the next page shows part of DNA molecules. Okay, and then it says label parts 1, 2 and 3. Okay, so the first question says you should label 1, 2 and 3. So the diagram, the number 1 in the diagram represents what? So number 1 in the diagram, it represents a nucleotide. Basically we know that the double helix molecule, DNA molecule, has what? It's a double helix structure, which means they are what? There's a phosphate group, there's a sugar group, and also there's a nucleotide, right? So these nucleotides, they are complementary to each other, which means that there's they always goes in pairs. There's a nucleotides. Basically we have 4 nucleotides. We have adenine, cytosine, guanine and thymine basically, right? So adenine goes with thymine, guanine goes with cytosine, right? So these are base pairs basically, and these are called nucleotides, right? So basically what they are doing is that they are the building blocks, which means they are in the building blocks of nucleic acids. Okay, so let me just emphasize on that. So many questions would say write me give us the monomers of the building blocks of matter, or give us the monomers of DNA, right? So basically you should know that monomers are the building blocks, and the building blocks of DNA, they are nucleotides, right? So in this case we're given what? We're given to label number 1. So number 1 in my case I would say what? So number 1 is adenine. Okay, so number 1 is adenine. Number 2, which in the diagram, number 2 in the diagram refers to what? Because number 1, the round one, is a what? It's a phosphate group. So, and then number 2, it's a what? It's a deoxyribose sugar. So number 2, it's a deoxyribose sugar. And then number what? Number 3. Number 3, it says which what does number 3 represent in the diagram? Number 3 represents what? It represents the hydrogen bonds that are bonding these two nucleotides. So basically what keeps all these bonds together without being displaced or being dispersed is that they are held together by hydrogen bonds, right? So what you need to know is that when you go deeper into the content is that each nucleotide has a specific number of hydrogen bonds that are linking these nucleotides, right? For example, adenine and thymine have they have 2 hydrogen bonds that are bonding the two molecules, right? Making them to be pairing, to be pairing also to form a structure, right? And then goes for your guanine and cytosine. They have they have 3 hydrogen bonds that are keeping them together, right? So for the label on number 3, would say that these are hydrogen bonds. All right, so let's move on to our next question. So our next question says, give the number of nucleotides shown in the diagram. Okay, so the number of nucleotides, as I've said in the previous example, that nucleotides are base pairs. They're always going in pairs, right? So for you to be able to give them a number of them, all you need to do is just to count how many nucleotides are there, and then you're just gonna count the number of them. Gonna double the number, basically. So, or maybe we'd say that you count how many deoxyribose sugars are there. So it's 1, 2, 3, 4, 5. There are five deoxyribose sugars, right? So it means that to each and every ribose sugar there are nucleotides, right? And you know that nucleotides are what? They are base pairs. They are base pairs that are what? Pairing together, held on together by what? By the hydrogen bonds. So in that case, how many will there be? So which means that if there are 5 deoxyribose sugars, then that means that there are what? There are 5 base pairs, which means there are 5 nucleotides that are shown in the diagram below. Okay, so let's write that down. So given the number of nucleotides shown in the diagram, okay, give the number of nucleotides. So there are what? There are 10 nucleotides. So number 3. So question number 3 says, name 2 places in an animal cell where this nucleic acid can be found. Right. So apart from what I've told you before, that a nucleic acid is found in the nucleus, there are multiple places that the nucleic acid is found. Right. So basically, it is found in the nucleus. First of all, where it's situated in the nucleus, in the nucleolus, and yeah, we know that in the nucleus there's free floating nucleotide and all that, right? So that's the first place you found the DNA. It's in the nucleus. The second place we found the DNA is where you find the DNA in the mitochondria of the human body, right? The nucleus, first of all. The nucleus. B, it's found where? In the mitochondria. Alright, so yeah guys, I'll try to explain this much further to you, because all I'm aiming to do now is to enable you to understand the topics into much more simpler concepts, because when time comes when you have to write exams, it's December, I know it might be frustrating. You need to be able to comprehend this content in a much more easily, or much more what can I say, predictable manner, or whatsoever way that you can be able to easily recall the information that I'm issuing you guys, you see. So hence I'm doing a step-by-step tutorial where you can see how I answer the questions relating to what the question entails, you understand. So let's move on to our next question, number 4. So what is the natural shape of this molecule? So the natural shape of DNA is what? It's a double helix structure. It's a double helix structure that it's a double helix structure that is called around the protein histone. Okay, so let's write that down. It's what? It's double helix structure around the histone. Okay, so let's do the last question. Let's do number 5, which is the last question. So what the last question says, it says draw a nucleotide with the nitrogenous base at 9. Draw a nucleotide with the nitrogenous base at 9. So basically what I'm gonna do is that I'm just gonna draw the whole entire structure. So to draw the nucleotide, so firstly we're gonna draw what? We're gonna draw the phosphate group. So we're just gonna write P. Just gonna copy how it's written there, how it's drawn there. So this is a what? This is deoxyribose sugar. What am I doing? Ish. All right. So 9. So these are the what? Just gonna represent these dotted lines is the what? Is the hydrogen bonds. Right, so this is how it's gonna look. Right, so this is the sugar. And then this is what these are our monomers. This is our nucleotide. This is adenine, and this is what? This is thymine. All right. So this is another deoxyribose sugar. So basically that is our short lesson for today. So basically, if you have any questions regarding the content of this video, you can just go down in the comments below and comment, guys, if you have any questions or anything of that sort. If you like my video, please like the video and subscribe to the channel if you say that you are a student that is doing maths and science and either taught in Xhosa or English, because what we do is that we do Xhosa and English tutorials in this type of videos. So yeah, guys, if you like, please subscribe and like the channel for more algorithm. So thank you very much, and also have a blessed one.
I think you captured it very well. There's no any better explanation than the explanation you have given.